All right, good afternoon and welcome back to FirstThought Focus. For those of you who are new to FirstThought, we are a life sciences focused expert network with a mission to bring subject matter expertise back into the expert network industry. FirstThought Focus is our virtual conference series that aims to showcase the breadth and depth of FirstThought's experts while also covering a wide range of topics that are relevant and important to the life sciences space. If this is your first time joining us, please be sure to check out some of our recent webinars that we've hosted. You can find the replays of those on our website, firstthought.io/focus. Our next event is going to be coming up next week on April 29th, and that's going to be with patent law professor Jake Shurkow, where we'll dive into the questions on how to understand intellectual property as it relates to cell therapies. As a quick reminder before we get started today, we will be taking questions from the audience at the end of today's discussion. If you'd like to ask a question, please submit them via email to hello@firstthought.io. Again, that's hello@firstthought.io. And with that, I'd like to introduce you to our moderator, Neil. Most of you have probably met him before, but in case you haven't, he is a veteran science journalist with over 20 years of experience reporting on technologies on drug development. In 2017, Neil authored A Cure Within, Scientists Unleashing the Immune System to Kill Cancer, which was published by Cold Spring Harbor Laboratory. I'll turn it over to you, Neil.
Hi, and welcome to FirstThought. My name is Neil Canavan. I'm broadcasting live from my fish tank once again. My guest today is Dr. Joshua Brody. Dr. Brody is the Associate Professor of Medicine, Hematology, and Medical Oncology at Icahn School of Medicine at Mount Sinai here in New York. Further, Dr. Brody is the Director of the Lymphoma Immunotherapy Program at the Tische Cancer Center Institute. This is also at Mount Sinai. And there he runs his own immunotherapy lab, and he applies his extensive knowledge of immunology to the developments of novel therapies, not only for lymphomas, but for breast and head and neck cancer as well. And these efforts also include his pioneering work in a modality known as Insitu vaccination. And hopefully, if time permits, we can discuss that a little later in our talk. Now, I would like to add just one more qualification to his long list of accomplishments. Josh served as the medical editor for my book, and there he is. And Josh, I just want to thank you once again. It was extremely valuable, your contributions to the work, and I mean, really, always. And to that point, I would like to, right before we get started, show you that the book just came out with this brand new spiffy logo for life sciences, LifeSci. And if you send your snail mail to Morgen, I will send you a signed copy so I can get rid of all these books that are in my apartment. All right, now, where are we going here? Oh, yes. So I
just have to say, of course, you're very welcome, but most importantly, 20 years of science journalism. I didn't realize you started this when you were 13 years old. That's really amazing.
It's true.
True. Very impressive. Right after your bar mitzvah, I guess you ran right into journalism. That's a mazel.
Well, I met someone at the dessert table, and things took off from there.
That's how it happens always.
Yeah. So, okay, I want to put up a graphic to just ground our listeners to the challenges that Josh faces every day. And this is lymphoma. This is the landscape. Now, Josh, I was wondering if you could break this down just a bit for me into the diseases where we have pretty good drugs, the diseases where you think there might be some pretty good drugs in the near term, and then those that are just thoughts and prayers.
Yeah, let me say, I think this is probably how a lot of venture capitalists look at lymphoma, and it's the wrong way because it does not the pie chart of unmet need, not at all. Chronic lymphocytic leukemia, I've been given specific permission to say the following thing by my now 100-year-old grandma who was diagnosed with CLL 36 years ago. She never got any medicine for that disease. It never affected her in any way, never progressed at all. So to see that giant magenta, fuchsia, purple, violet slice doesn't tell the story of unmet need. CLL is a very common disease and lots of folks go after it. But number one, It's not as morbid a disease as some of these and the rest of the pizza pie. And we have amazingly good medicines between antibodies, chemotherapy, BTK inhibitors of several types, and BCL2 inhibition, not to mention more recent PI3K delta, et cetera, et cetera. So, the unmet need of that is more like the lavender slice, the mauve pizza pie slice of hairy cell leukemia. So, the unmet needs here are of these gigantic turquoise DLBCLs. 1/3 of those patients are still a profound unmet need. And then actually we can lump the follicular orange and marginal zone, whatever, periwinkle, I don't know. We can lump the two of those pizzas together and just call them indolent NHLs, as well a couple of these others are. And of those indolent NHLs, 75% of those people have great options and do well and live long, pretty healthy lives. But a quarter of them, so a quarter of that pizza pie, the two slices together, have this bad version of indolent lymphomas. We just call them the early relapsers. They get standard chemo and relapse within a couple of years, and that's a profoundly unmet need. So 2 unmet needs: a third of the DLBCLs, a quarter of the low-grade lymphomas, and then almost all of these T-cell lymphomas, the green there, are almost all an unmet need, or certainly 2/3 of them at least are the unmet needs. So yeah, it's harder to convey the pizza pie of unmet need, but it's certainly amongst those 3 foremost. Yeah.
Okay, great. Thank you for that. Now, for our listeners, this is how we're going to proceed today. This was going to be like a CAR T versus BiTE seminar, but I've shifted focus because of AACR. And I think it would be remiss not to have Josh know so much that we might as well just go into some of the things that we saw at AACR. The way I'm going to proceed is with CAR T, abstracts for CAR T first, then T-cell engagers, and then this will follow into other cell types like tumor infiltrating lymphocytes and gamma delta cells, then onto immunotherapy activators, that would be STING. Then we're going to into ADCs, antibody drug conjugates, which used to be a thing and now they're back and now they're a thing. And then monoclonals for lymphoma, and then finally small molecules for lymphoma. So to dive right in, this first abstract, number 1549, this is by a company called Arsenics. Now, Wall Street must really like this because that week they scored $115 million Series C. Now, this is a fairly exotic technology to me. I'll describe it briefly for Josh, and you can let me know what you think of it. What they've done is they've taken the idea of a CAR and they put a generic post on this thing. And then they developed another drug that attaches onto the tumor cell and has this post sticking out of it. Now this post attaches to that post. That's the drug, or that's the procedure. Then the reason they've been doing this, because this thing can exist in your body and float around and attack this one. But if you lose that target, they can target with something else, and then it'll go to that one. So it's a generic CAR T cell. And theoretically, this will also tone down the adverse events. Seems kind of cool. Josh, any ideas?
There's 2 points of it. One, sort of the on-off switchiness of it, and the other, what I call Flexicarness of it, Flexicar of it. And both of those concepts have some excitement. Excuse me if my office makes a lot of noises. And Flexicar is elegant and great. Some of the companies have just been thinking about using Flexicar in the context of just normal FDA-approved antibodies. So I can just give my patient rituximab, cetuximab, Herceptin, whatever, and have my Flexicar work with already, so you have one CAR product and then all these FDA-approved products. And then these guys are, individualizing it to their BCMA recognizer. Flexicar is great. Neil, I'll push back on the idea that, that you said, and what these guys, they're not the only company. There's, I think, 3 companies at least working on this Flexicar concept. And the idea that I'm going to give you the CAR with the BCMA targeter, and then when your tumor loses BCMA and does antigen escape, then I'll just give you a new targeter. Now, the problem is the CARs are not going to be there like they were on week 2, 3, 4 when you got them the first time. So not simple, we'll salvage the CARs that were already there, I don't think. They'll certainly be, either or gone or manyfold less, and whether we can resurrect them just by bringing them another ligator, another recognizer, I'm a little skeptical. So the Flexicar concept is great for practicality, maybe get one FDA CAR approved and then just add it with multiple antibodies. It's an elegant concept, but I don't think it's going to be the solution to antigen escape. And we can talk more about antigen escape a little bit because that's something else we published on pretty recently. In terms of the on-off switchness of it, on-off switches of CAR T cells, it is an embarrassment, I think, to our field that we do not have these in our patients really yet, or not FDA approved certainly, or nowhere near that. And I don't like to take the sides of companies that eventually didn't do great, but I don't want to side with General Custer from the war that he didn't do well in. But Bellicum had a great on-off switch based on the Baylor, Malcolm Brenner and company technology. That technology was in human beings, not in CAR Ts, but regular T cells. In 2010, published New England Journal of Medicine 2011. It was a great on-off switch, and just practical things, I think, not the science, I think, didn't make it go forward. So I believe strongly we should have on-off switches in all of these. You wouldn't drive an automobile that did not have a brake pedal in it, and certainly safety is still a primary unmet need of all CAR T cells, pretty much. Some even more than others. And so I don't know if this is the optimal off switch, because I honestly just don't know the kinetics of how quickly this approach can actually turn them off just by withholding the ligator. We need the off switch to turn off the CAR T very quickly. Some of the small molecules have very quick kinetics and can do that very quickly. So if they show us some data that this withholding the adapter piece can quickly turn off CARs, then I'd be excited. Otherwise, I would not be excited for its utility as an on-off switch.
So you mentioned Antigen Escape. Bluebird just had a CAR approved in multiple myeloma. It's not going to address that problem, but good luck to them. Godspeed. I know Marcelo Maz recently published on a dual targeting BCMA CAR to address this idea of antigen escape. You said you published recently. Give me your thoughts on how to do this.
Well, I mean, the basic approach is very reasonable, and then I'm going to pick holes at it in a second because that's what we do. But the basic approach is go after a second target, either sequentially or in parallel. And so I think Marcelo's approach, if I remember correctly, is the CD19, CD20, quote, unquote, bispecific CAR. We have CD19, CD22 CAR also in patients. So conceptually, it's good and should improve, partially solve the problem of antigen escape. Here's a couple of problems. Problem number one, sometimes when a 19 cell loses 19, it doesn't lose 19 by itself. It loses its whole sort of B cell differentiation program. And so it can lose 19 along with 20, along with 22. And there's some nice data about this. I don't know if that happens frequently or half the time or a third of the time, but it is a problem that when you lose 19, you lose other things, number one. And number two, the problem is, and also I think Marcella and colleagues, that might be a different paper, was going after BCMA and had the April ligand.
Yes,
CAR,
that's
so
the one.
both TACI and BCMA at the same time. But just giving that as an example, it's not common that we have two safe targets. So, B-cell lymphomas are, shooting fish in the barrel in terms of targets. We're very lucky in these cancers I take care of to have a lot of good surface targets. But, the goal of CAR T is not to just do better in DLBCL. I wish that was the whole goal, but to go into prostate cancer, glioblastoma, mesothelin expressing tumors, and so forth. And I don't know if we have a second target in those guys. So solving the problem of antigen escape can't just be, go find a second target, because we're lucky to find a single target most of the time. So I'll only give the one-liner about our pitch about how we can try to prevent antigen escape instead of treating it after it becomes a problem. And our pitch is it's a very inelegant concept, as a lot of my concepts, which is that T cells in all of us did not evolve just to kill the on-target cell. They evolved to kill the on-target cell, and just to be safe, even though it's inelegant, to also just kill the neighbor. Because sometimes when you got a lot of bad guys, sometimes it's a bad neighborhood, and we don't want to kill the whole body, but just to kill the neighbor would be how to play it safe. That is how our T cells evolved. If my throat has a lot of virus in it and one cell doesn't happen to show the virus, it probably got exposed to. Just kill the neighbor. So this thing we call bystander killing just the next door antigen negative cell, even not 1,000 next door neighbors away, but just the next door neighbor, is something that's probably happening in all of our T cell therapies already today. And the thing is, we also think we can increase bystander killing because we have a pretty good understanding of how it works. It works mostly by a fast ligand and fast ligation of that neighbor cell, and we can increase fast signaling. And we have things in the clinic like IAP inhibitors, IAP, inhibitors, and also maybe soon, maybe we'll have c-flip inhibitors. These increase fast signaling. They would increase bystander killing. They would slightly increase the sloppiness of a T cell killing its target, killing the neighbor even more effectively, and might prevent antigen escape in that way. So that's been about our pitch that we published a couple months ago. And so both approaches, I think, reasonable, going after a second target, but when we don't have second targets, just combining with some of these small molecule inhibitors that might help to increase bystander killing, make the whole thing a little dirtier, but really get the antigen negative cells as well.
Okay, next abstract, this is CT007. Ooh. This is by Orca Biosciences, and this is a bispecific CAR T CD19, CD20. The secret sauce here is apparently they're using naive memory T cells in the bag. So my two questions. First is, why would you use my naive memory T cells as opposed to any other? And the second would be, what do you think the most efficient way is to deploy a bispecific CAR? Would it be 2 CARs with 19 and 20? Would it be 1 CAR that has 19 and 20 on one tether? Or would it be 2 CARs with a construct 19 and 20 separately within the same cell?
I think you were also asking me about, whether the naive slash memory cells are critical here. And I think in concept, it's great, but in practice, donors have what they have, and we cannot probably enrich enough to make every patient give us the T cells that they want to give us. And if they've had 4 lines of prior chemotherapy, you're right, their T cells may not be as good. They may not have as many fresh naive T cells, naive or memory T cells. The concept being that they will last longer after we reinfuse them and also tolerate the manufacturing better. But I don't think it's an actionable thing. I think it's a correlate, but not something we can enrich for always. In terms of the best way to do it, for example, 1920, all things being equal, and all things are not equal, but all things being equal, it'd be great maybe to have them on the same cell to keep as many cells activated. Even if the 19 cell didn't see 19, still it could get activated, the same cell by seeing 20. But my understanding of packaging these things into gamma retroviruses is it's not a technical no-brainer. It is harder to get more genetic info into one gamma retrovirus or lentivirus, and that you don't get perfect expression of the anti-19 and the anti-20. It's a little bit of zero-sum game. You lose a bit of the efficiency of making great CARs on all the cells. So technically speaking, I think actually maybe having the 19 and the 20 on their own separate cells and having them both be highly efficiently transduced by the virus that's making the CAR T might be better. But I think overall they'll be more similar than different. And again, I endorse the concept of going after a second target, but some limitations from what I mentioned before.
Well, that answer might inform the next question, and this is Late Breaker 028. This is a company called Senti Biosciences, and they did a logic-gated CAR on an NK cell. So the idea being, they're putting in 2 targets, FLT3 for AML, and a healthy target of choice that will be on the healthy cell. So the idea is, since FLT3 is also expressed on healthy cells, that if the CAR comes up to your healthy cell and finds both targets, it'll turn off.
Absolutely. So elegant, and the innovator or leader maybe in the field, Michel Sadelain and his group, were publishing on some of these Boolean logic CARs a few years ago already, Nature Medicine, I think, and conceptually incredibly elegant in terms of, this is not ready for prime time. I don't think that's anyone's expectation. But why couldn't this be the future? It's just a matter of, will we be able to think of the good healthy targets as well, and make the Boolean logic selector, really quite sensitive, that it really turns off when it sees a healthy cell and really stays on when it doesn't see the healthy cell. And we should just say, the example here is about a cancerous myeloid cell versus a healthy myeloid stem cell, because that's been the problem in AML, is just all the targets are also on healthy stem cells, it seems like, as well. So that's a great example, but an even simpler example is just pick like EGF receptor on many of these lung cancers, on a lot of other tumor types as well, but on every skin cell in your body, fry the lung cancer, fry the skin. My father's a dermatologist, that would be bad, he tells me. So yeah, but there should be so many easy ways there to target, EGF receptor on lung tumors and spare healthy tissues. So it's not like this is limited to AML and sparing healthy blood stem cells. Lots of application of this, conceptually fantastic, but again, this is not something that's won or lost in concept, it's won or lost in implementation. So I definitely am enthusiastic about the concept. I hope that they can implement it. Yeah.
A more general question related to this asset would be an NK CAR. So why would you, what's the advantage of putting a CAR on an NK cell as opposed to the T cell?
I'll try to answer the NK question, but let me answer just the general allo questions a little bit first built in there, because we're talking here about allo NK cells, which is that, you know, Neil, you and I have talked about this a number of times over the years. Over the years, I've always been a little bit negative about allo CAR in general, because I think they address problems 6, 7, and 8 of CAR T cells, but not problems 1, 2, and 3, which are, you know, efficacy, or efficacy and safety and usability. And, you know, they're addressing problems like, you know, feasibility of the patient. And they say they're addressing the problem of cost, but that's silly. They're not addressing the problem of cost. They will charge quite a bit for any FDA-approved product. It'll be 2% more than last year's medicines, just like every year. But they are, you know, trying to address the make things off the shelf for a quicker time, turnaround time. Because I used to say, and this is where I've become a little kinder to the allo CAR field, I used to say, oh, we always have 18 days for, you know, kite to turn these around. It's not a big deal. But certainly we've seen a lot of patients where 18 days is a make or break problem, and having something ready today would make a difference. So I'd be a little less mean about that now. You know, all in all, for that little benefit of an allo CAR, you know, what's the downside? The downside is I don't believe we'll ever solve the main problem of allo cells. So allo cells have two problems: the CAR attacking the allo T cell attacking the person, graft versus host disease, and the person attacking the transferred allo cell rejection or lack of persistence. The graft versus host disease is pretty solvable. Lots of groups solving it in different ways. Many of them CRISPRing out the endogenous T cell receptor so you won't attack the person. The CAR transferred allo CAR T won't attack the person. So it's pretty well solvable or solved. But the person getting rid of the allo transplanted CAR NK or CAR T, difficult to solve. Very, very difficult to solve. I mean, you can solve it temporarily for a few weeks. You give them some chemotherapy and they are T cell depleted and they won't hurt the transferred CAR, but hard to solve it long term. So I think that's generally a problem. And therefore, I think overall, the downsides of allo certainly don't match up to the upsides of it, which is the off-the-shelfness. There is one other upside to, which is finally getting to your question, one other upside of the allo cell, which is if you have a really young, like an induced pluripotent stem cell derived NK or T cell even, and Fate is one example of these. I think Precision is sort of on that model. Then the more long-lasting that cell is in the culture, in your petri dish, the more genetic manipulations you can do to it, and you can start to do a lot of cool genetic manipulations. And you know, to get these logical, you know, plus minus CARs in there, they have to be in there high efficiency. You don't want to put in the plus without the minus or something like that, or have one be expressed and not the other. So in this sense, for these fancier genetic constructs where you really need more than one thing to be in there almost all the time, that's where you do kind of need an allo cell. So you have time, you know, you don't have 18 days to make it since the donor gave it. You have all the time in the world to make it over 5 years and then just have batches to hand out off the shelf. So I think that these Boolean and fancier genetic constructs in general will benefit from the allo space. So yeah, that is one great purpose of the allo CAR T or NK. In terms of T versus NK, I have to say I'm a little unconvinced of the benefits of NK. Their primary benefit is you don't have to solve the graft versus host problem because they don't have an endogenous T cell receptor to recognize the recipient as foreign. So they kind of solve the first problem, graft versus host. They solve it by themselves, but they still haven't solved the, you know, how does the person not reject the CAR NK? So the benefit is small, I think, of NK over T cells, and whether T cells attack better cell per cell. I thought that we think that they do. So overall, I'm not overwhelmed convinced of the benefit of NK. I think T cells.
Okay, all right. Moving on to a dear old friend, an ancient friend, IL-2. The first, arguably, well, second immunotherapy drug after Coley's toxins, I guess. This is abstract 1512. The company is Synthekine. And if I understand what they've written here, they have rejiggered the IL-2 receptor on their CAR so that it will only accept the synthetic IL-2 drug that they are going to sell along with it. And the idea being that this cuts down on the toxicity and also targets it directly to the proliferation of CAR T cells. Lots of people are trying to attenuate the toxicity of IL-2. Your thoughts on anybody succeeding?
Yeah, well, I mean, this doesn't totally solve the problem of IL-2 toxicity, because still my own endogenous T cells will still have my normal IL-2 receptor and can be toxicity associated with that. But overall, I just think this is just another gas pedal. Our best is to have a gas pedal and a brake pedal. This will certainly turn on the T cells. When you stop giving the synthetic, mutated IL-2, will they turn off quickly? I'm a little suspicious that they will not turn off quickly. They won't keep proliferating as much as when you have the mutated IL-2 there, but I don't think they'll turn off quickly. So I think this is probably a pretty good gas pedal, because IL-2 is the most famous gas pedal for T cells. I don't know if it's the best gas pedal, but it's certainly a very well proven one. So it's a very good gas pedal. I'm not convinced that when you just take your foot off the gas that these cars will stop. The car will still idle. So I think it's an elegant gas pedal. Honestly, I think you kind of need these gas pedals with a brake pedal as well.
Okay. Moving on to where are we? Late breaker 026. Yeah. Oh, this is IL-12, now a different cytokine, IL-12 this time. This is in a CAR T. This is from Refuge Biotechnologies as well as ESSA Pharma in the collaboration. Let's, in general question, the use of IL-12, what are they getting out of this, and do you think it's worth it?
So, IL-12 has been around for 30 years, certainly, but we've been trying it in patients for 15 at least. We've been trying in different contexts. Certainly the NIH, world leaders in adoptive T cell therapy have put IL-12 into different types of T cells, transgenic T cells and CAR T cells. And other folks have just been injecting IL-12, the protein, IL-12 plasmids. There's an ongoing RNA, since RNA is very sexy this year in the era of Moderna and BioNTech, injecting IL-12 RNA along with a couple of other cytokines. There's clinical trials ongoing with that for local injections, sort of a type of Insitu vaccination. So IL-12 is unquestionably a potent T cell activator, unquestionably. And the problem with all those things I just mentioned has been delivery is a little bit toxic. IL-12 kind of doesn't stay where you inject it, or if you put it into a CAR T cell, it doesn't just stay in the exact locale of where the CAR T cell lands at the tumor. IL-12 goes a bit systemically. Certainly the downstream of effects of it goes systemically. So you kind of put these people into a cytokine release syndrome situation, or a septoid type of a situation, and it's a toxic therapy. And so that's been a limitation of IL-12. So I think both this and maybe the next one we're going to talk about, which are these exosomes packaging IL-12, both trying to solve the same problem, trying to keep the IL-12 more localized. It is, again, a nice concept. I'm not quite compelled about the implementation of the concept, whether we've really achieved the goals of the concept to keep the IL-12 local. Worthwhile concept. Not a lot of evidence here of success of the implementation, but we got to try to succeed, I guess.
You just alluded to the next abstract, which is CT114. This is from Kodiak Bio, and yes, it's an exosome with this entirely different type of cell than we've been discussing. You already responded to the IL-12 part of it. Just for perhaps some of our audience is not familiar with exosomes because they're not really prime time yet. What are they, and why do you use them? Why would you want to use one?
Not a cell type per se, really a cell fragment almost. It's just a couple of things away from being almost just a lipid bilayer, tiny little bit to package something. Exosomes are something that all of our cells are accidentally making, not as part of their primary function, but certainly some dying cells can release exosomes, and cancer cells that we study this the most can release a lot of exosomes. Usually, not surprisingly, they are sort of immunoregulatory or immune inhibitory exosomes. And it just depends on what is in the exosome that it releases. So it's just a little fragment of a cell that gets released. And some of them can, one of the famous ones that we studied have been fast ligand expressing exosomes. So if the tumor cell has a lot of fast ligand on its surface trying to keep fast expressing T cells away from it, it would be toxic to the T cell to come near, then that's a local weapon against approaching T cells. But you can turn that into a distance weapon against T cells by just taking that surface fast ligand and releasing some of the plasma membrane to become an exosome, and it'll go kill approaching T cells downfield. So, really it's just a packaging device. And, the belief here of this group from Kodiak Bio is that it can be a more effective packaging device than just other lipid bilayer nanoparticles. But honestly, I think it's just another nanoparticle with which we can package a certain amount of anything, in this case IL-12, into a little protective layer and release it, hopefully just at one site and not have it go throughout the body.
Got it. Got it.
It's a local
Now we're going back to T cells, this time tumor infiltrating lymphocytes, T cells. The company here is Iovance. They are the unquestioned leader in this space. And this abstract has to do with CLL and small lymphocytic lymphoma. So my question is this TIL a thing for lymphomas?
Let me first say the nicest things about TIL, which is these guys are the leaders in the space because this is a long tradition passed down from the NIH and Steve Rosenberg and Mark Dudley and Nick Restifo to multiple biotechs, and before Iovance it was Lion, and all admirable stuff because this work 22, 23 years ago and then first published in Science in 2000 in melanoma patients was transformational. It's first and foremost an example of a concept which proves a school of thought, even if it's in and of itself difficult to implement. So TILs, this is not the post-Nobel Prize era. This is the era of nobody believed immunology or certainly cancer immunotherapy was relevant. This is 2000. This is the year Gleevec came out. And everyone said, that's it, Gleevec, and then just make a Gleevec for every cancer, and that's how we do cancer. Get all these silly immunotherapists out of my hair, ask them to stop requesting grants because Gleevec is the only thing between now and the end of cancer. And it was a small party of us that disagreed. And Neil, you just finished your bar mitzvah around that year, and you were a part of the burgeoning school of thought that T cells can kill cancer. And these papers by the NIH, Rosenberg, Dudley, Restifo, that became Iovance technology down the line, proved the school of thought that T cells can kill cancer. It was just a belief until then, and then it became a fact. And that fact, the pictures of the gigantic melanomas that melted away because of this TIL therapy was inspirational for a generation of young, recently bar mitzvahed, burgeoning immunotherapists. So
yeah, but what about lymphomas?
So melanoma, definitely, and why? And then I'll just say cervical cancer, where also Iovance has got the most progress, should be a no-brainer. I mean, where will TIL therapy work? It'll work in a tumor where there's a bunch of anti-tumor T cells already in there. And why would there be a bunch of anti-tumor T cells? Because there's plenty of targets. Melanoma has plenty of targets because it is a very highly mutated disease. Cervical carcinoma has, I don't know about plenty of targets, but some great targets because it has HPV viral targets, which T cells are real good at. And then maybe lung cancer conceptually. And certainly the NIH has done a lot of, NCI and Steve Rosenberg and team have done a lot of different TIL therapies for different cancers. And in all that time, melanoma is the great one. Cervical cancer is pretty good, we think. And we haven't seen evidence from other ones that it can work in a significant proportion of patients. SLL, CLL has on the lower proportion of tumor mutational burden, so we think has less anti-tumor T cells floating around in the tumor. So although I wish that this therapy that I love and this disease that I treat all day would match up, I'm a little skeptical, and now I'm making fun of some of my idols because I'm a little skeptical that SLL has a great place here.
Okay, and moving on to another type of T cell. This is a gamma delta T cell, which are not in abundance in the body, and this is the CT034 and by IM Check Therapeutics, and Patricia LaRusso is on this, so it must be something there. It's a monoclonal antibody. It's targeting a target I've never heard of, butyrylphyllin 3A, and the idea is to activate gamma delta cells. So if you could just touch on briefly, why would you want to use a gamma delta cell, and have you heard of this target?
So I am also, like you, Neil, not a butyrylphyllinologist. And it has some upregulation on some tumor types, not extremely specific for any tumor per se. Not only is Patricia an author here, but so is my very good friend Aurélien Marabel, who leads an incredible program at Gustave Roussy, the phase 1 immunotherapeutics program. And as you know, Gustave Roussy is kind of the MD Anderson plus Sloan Kettering of Paris, so they could do an incredible amount of great work. But I have to be honest, I can't speak expertly, even though I knew we were going to talk a little bit about this. It's still beyond my expertise of how great of a target butyrophilin is, because those are hard questions to ask. I can show you how much of it is expressed on different tumor types, but I can't tell you how clean it is. Those are harder questions to ask, and I don't have the answer.
And the gamma delta cells?
Gamma delta cells conceptually, the nice thing about them is, again, they should be a little bit less GVHD inducing. So certainly you can infuse allogeneic gamma delta cells more safely than you can infuse the common T cell, which is alpha beta T cells. So the concept there is reasonable. I don't think that they are more potent at killing cancer cells cell per cell, but they have that initial safety aspect to them.
Okay. Now moving on to a bispecific. This is a CD3, CD20. This is by Genmab. They know, they've been doing this a while. They must know how to do it. But my question here is, when you have Rituxan, why do you need this?
Oh, that is embarrassingly what I thought 8 years ago or so, before we had good data of them. So bispecifics are just better. Here's why they're better. I mean, it's not a question, it's a drastic effect, because all of these best bispecific trials, and epcoritamab is one of 6, really. We have 6 companies, or actually 6 products. Genentech has 2 of them, so maybe 5 companies and 6 great products. 2 from Genentech, 1 from Regeneron, 1 great one here from Genmab, Xencor, and certainly others coming down the pike. Bispecifics, just to say this bluntly, are the future of lymphoma therapy, and that is an opinion, but it's also a fact, because I'm smart enough that my opinions are facts. And bispecifics work when rituximab fails. And the reason why: rituximab recruits a natural killer cell, or maybe a macrophage, to kill that tumor cell. But number one, maybe an NK cell is not as potent. And most importantly, an NK cell does not get highly activated just by seeing rituximab. It gets moderately activated. The Fc gamma receptor activate that NK cell moderately. But these guys, they bind to the lymphoma cell by CD20, they bind to the T cell by CD3 and potently activate them. That's a concept. Forget the concept. Here are the facts. All of these patients on these 5 different platforms that have had awesome response and complete remission rates, both in aggressive lymphomas, DLBCL, and low-grade lymphomas, even a bit more so. Just the low-grade lymphomas, multiple of companies here with response rates from 60 to 80%, and complete remission rates above 50%, just unparalleled. There's no other medicine in non-Hodgkin's lymphomas that have had these numbers ever. So unparalleled early efficacy and pretty good tolerability. There are some problems with cytokine release syndrome, not as bad as CAR T cells. And I think that those cytokine release syndrome problems have maybe been solved or are being solved just using a simple common sense thing, which is up titration. You don't give the full dose on day 1, 1/10 of the dose, then 1/3 of the dose, and then the full dose. And it seems to have mostly solved that problem. So epcoritamab is just particularly exciting because it's really sort of number 3 or number 4 in line, but they are trying to leapfrog ahead of the others because they are the first ones to go by a different route of administration. Sounds so simple, but all the other ones started as intravenous, and epcoritamab is starting as sub-Q. And it seems like just by the slower infusion into the person from a depot in the subcutaneous space, it's been safer. It's just basically giving the medicine slowly over time, I think, is why sub-Q seems to be safer. So epcoritamab a little bit leaping ahead, and also starting itself almost right away. They finished the phase 1 trial of monotherapy, but now jumping right into this combination trials. And that's how you get from 3rd line FDA approvals into 2nd line and 1st line approvals. So all the bispecifics, fantastic. The practical implementation of how epcoritamab is doing it, super exciting. I mean, this abstract doesn't tell that full story, but bispecifics for lymphoma, great. For myeloma, probably great. We even have some for, outside of my expertise, but even for prostate cancer, I think we have one company, Harpoon Therapeutics, with a pretty promising bispecific, and a few others coming down the pike as well. And epcoritamab, high amongst them for lymphoma.
I just want to add a little bit of color for our audience as far as Rituxan. Josh trained with Ron Levy, and he was the one who developed Rituxan.
Let me just say, whenever Ron hears that said, he always kind of pushes back and said, I helped to develop rituximab. Really, what he really did was developed monoclonal antibodies for lymphoma, anti-idiotype antibodies, and that spurred the development of the company, Biogen IDEC, or IDEC, I guess. And then the board said, this is too complicated, customized antibodies for everybody. Let's just make CD20 across the board. And the story was pretty good. It built South San Francisco as a town. We now call
Wow.
that town Genentech, but it's a beautiful town because of that decision. It was a good decision. But I got to say, in 1994, when they were making that decision, the belief was if you ablate someone's B cells, they will be a bubble boy or bubble girl. They will have no immune system. Turns out you can live surprisingly well without your B cells for many years. A little infectious risk, but fairly small and surprising. Neil, I think from the hints of the abstract you told me, we're moving away from CAR T cells, but maybe I'll just mention one that was at AACR, and I don't have the abstract in front of me, but it was kind of promising because when you show that pizza pie, I mentioned the unmet needs are some DLBCL, some of the chemo-refractory low-grade B-cell lymphomas, and then I said that green slice, the T-cell lymphomas broadly are all unmet need, just about. There's a couple of T-cell lymphomas we do okay with, but T-cell lymphoma is the biggest unmet need. And with all this CAR T talk about helping B-cell lymphomas and helping plasmacyte cancers like myeloma, what's happened to us, getting rid of T-cell lymphomas and T-cell leukemias, and the progress has been slow. And we have a little bit of progress now. I think at AACR, I think this group Gracell, if I'm pronouncing them right, Gracell, presented. Actually, it was really a follow-up, but the follow-up's important in CAR T cells because transient remissions don't count for anything, but durable remissions really do. And they had a follow-up just of their first 6 patients, I believe, and I think 5 or 6 out of 6 had complete responses to their anti-T cell CAR T cell, and so it's targeting CD7. And because we want to avoid this thing called fratricide, where the CAR T cells could kill their brother CAR T cells because they all express CD7, they knocked out CD7 from the CAR T cell, so it can't be killed by their brothers. And so they CD7 targeted, CD7 themselves knocked out, CRISPR'd CAR T cells from Gracell. We're doing a preponderance of remissions in patients with a T cell acute leukemia, a horrible disease, not a common disease. And the update this year was that they had a number of patients with remissions lasting more than a year. Certainly many of them were more than 6 and 9 months, and some of them more than a year. And that's great for T-cell leukemia, a rare disease, but T-cell lymphoma, a much more common disease. Again, as you showed, it's 10-15% of lymphomas. So yeah, that would be our first foray into really making these CAR T revolutions help our patients with T-cell lymphoma.
Great, thank you for that mention. I do need to mention also for an audience that we are open for questions now. Josh and I know each other pretty well, and we could talk all day long, which, yeah, we're still gonna do that. But I'm gonna, send your questions in if you have any. Now we're gonna do something, this is CT110. Now be nice, because Jason Luke is on this, and he's a nice person. Sting, this is by SynLogic. A number of people have Sting. Go ahead, hit it.
Well, Sting agonists, when we started doing the clinical work 4 or 5 years ago, based on some preclinical work, but based largely on this beautiful work from Tom Gajewski's group, this beautiful animal work, we were so excited because, as you know, activating dendritic cells and some other immature myeloid cells that present antigens to T cells is part of our shtick, many people's shtick, ourselves included. And we thought Sting was just in the animal models looking so amazing. And yet we've had a few, Doro and Merck 1454, that have been lackluster. It's not that they did nothing, but they didn't, certainly wasn't a home run. And this is now years into those trials. And these were big phase 1 trials, many of them already combined with anti-PD-1, the Merck combined with pembrolizumab, of course, for many types of cancer. And, we didn't see any home runs. So the question is it a failed concept or just a failed implementation? Was the Sting agonist not being sufficiently delivered? Was it getting neutralized by immunoinhibitory environments in the tumor? And I don't really know the answer. And unfortunately, the animal models may not answer it. So we have to be helpful here for this abstract about SIM B181 but still, this abstract is pretty early. What it's showing is safety of this Sting agonist, and we still are looking to efficacy. So we should keep our eyes open to it. Hasn't solved the answer yet.
Okay, this next one is an ADC, and it's sort of like what you just said with Sting. Is like when ADCs first came out, they're like, oh, this is really cool. Quickly for the audience, an ADC is an antibody on one end and a poison on the other end, and this is it targets the cell. The cell takes in this construct. The linker between the antibody and the poison is snipped. Poison is released, kills the cell. You don't get systemic toxicity. The poisons they're using, you could not use without doing it this way, or you'd kill somebody immediately. This is a CD70 ADC by, there was no company named on this abstract that I can tell. So, first you like ADCs, and first you like this target, CD70.
Well, ADCs have, to some degree, delivered on their promise. I mean, it's not a concept, it's a practice. It's standard therapy for Hodgkin's lymphoma, a bunch of T-cell lymphomas, breast cancer, and now certainly bladder cancer, first solid tumor from the Seattle Genetics, which is a cousin of this company, a cousin of this product, sorry. Real efficacy. And, so far, the poster child, I would say rituximab vedotin, the CD30 targeting ADC, now standard frontline therapy for Hodgkin's lymphoma combined with chemo, and also used in later lines of therapy. And in breast cancer and in bladder cancer, second and third line therapies. So they are delivering on their promise, even if they have not, cured all cancer like we kind of thought they would. And I'm sorry, we have other examples as well. Polatuzumab vedotin, CD79 ADC, using the same platform as this one here, the CD71. So ADCs, no question, fantastic. They haven't worked everywhere, but they've worked in disparate places and with good potency and good safety. So they are a thing, and they are great in lots of, they're not niches, but small places still. They haven't solved lung cancer yet or pancreas cancer yet. So they're great. And what about this target CD70? Actually, we've been looking at this target for more than 10 years clinically. So up until now, it's been a little bit lackluster, probably because it's not a very clean target. So going back here to this, mouse model targeting xenografts, it's promising, but the clinical data so far hasn't been here yet. So maybe it's just that we haven't used it in the clinic in the right diseases only, and this is targeting cutaneous T-cell lymphoma. So maybe we just need some more CTCL, anti-CD70 antibody drug conjugate clinical trials. The places we've tried it, like kidney cancer, this exact therapy, hasn't been a home run.
All right, but now we want to move on to, this is from Arch Oncology, abstract number 954. The target is CD47. They want to use it. They say it's efficacious in preclinical models of lymphoma. So 2 questions. Do you like the target? 2, are preclinical models of lymphoma adequate?
Tough questions. CD47 contentious over the past few years. It was developed in the lab right next to me at Stanford with these super geniuses, Mark Chow and Weissman, and the company 47, I think had mixed results. As a monotherapy, lots and lots of patients, not a lot of signal. So hard to call that a success. The success that they had, I thought, was they had 2 pieces of success. The first was in combination with rituximab, which makes sense. I mean, really, I don't think the thing, what is this, blocking a don't eat me signal, but it's not like the immune system was about to eat in the first place. So I think you need an eat me signal like rituximab combined with this therapy. And the rituximab plus anti-CD47 New England Journal of Medicine 2017 paper was pretty promising. It was just a small group of patients for DLBCL and follicular lymphoma. And when they expanded the cohort to get a few more, they presented that at Lugano meeting a couple of years after, and it wasn't quite as good as other therapies we have for those 2 diseases. And then they've had this exciting bit of progress with acute AML, acute myelogenous leukemia, in combination with methyltransferase inhibitors. And I got to say, I don't understand why that combination worked well, but it looked promising. And then 47 got bought up, and I don't know the direction it's going now. Overall, I think this is a saturated target in that we have 5 companies going after this target. And, when you're doing a complex thing like making something work, like IL-2, turning something on, there's lots of ways to turn things on. But I have to say, there's only a few ways to turn things off. Inhibitory antibodies, whether it's pembrolizumab, nivolumab, or the other 7 versions of those, inhibitory antibodies all work the same. So there's blocking a thing is straightforward, and this is just blocking 47. So I'm a little skeptical how the 5 companies could distinguish themselves and how preclinical models at this point could get somebody to leap ahead of the companies that are already 5 years into this.
Okay, we've had 2 questions come in. The first being, what are your thoughts? Oh, Morgen, you want to do these?
Sure, Neil. Hi. Hi, Dr. Brody. So our first question that came in from an audience member is, what are your thoughts on Insitu vaccination as a follow-up in indolent non-Hodgkin's lymphoma, and also in any other cancers that you can think of.
So Insitu vaccination is something, as Neil said, very near to our hearts here. It's something we focus on a lot. We were very lucky to publish something in Nature Medicine a couple of years ago about some great results in indolent lymphomas that was using Insitu vaccination as a monotherapy, just for the uninitiated or whatever. The whole concept is just all oversimplified, injecting some immune stimulants into a tumor to get the whole immune system to recognize that as being like a pathogen. And as Neil, as you said, William Coley was doing this 115 years ago already. And we've advanced a little bit in our understanding, so we think we can do it a bit better, knowing which cells we have to activate with these immune stimulants that we turn on. And so we had some good results in indolent lymphoma. The key difference of our approach to some other approaches was that when you inject an immune stimulant, it has to land on an immune cell that can hear that stimulant, that has the receptors to be activated, to get excited, and then teach the immune army to go recognize this tumor throughout the body as a bad guy. And that critical cell that hears the immune stimulant and instructs all the immune soldiers to fight cancer is called the dendritic cell. And the big difference now is that we have ways of increasing dendritic cells in our patients' whole system and in their tumor specifically. Our favorite way is called FLT3 ligand. FLT3 ligand is mostly been put forward by Celldex over this past 10 years now, by Immunex in the late 90s. And the attention to that has been sparse because the intellectual property is a little bit old on that FLT3 ligand. But I think it's, some of folks' work, ours and some other great teams like Nina Bardwaj and Sandra De Maria and a few others have brought FLT3 ligand to so much the attention of big pharma that I am certain that you will see other versions of FLT3 ligand or things just like that coming out in the next couple of years. So broadly, I believe in Insitu vaccination strongly. I believe that getting the dendritic cells will make it work. And then the question about other cancers, our follow-up trial is combining this Insitu vaccine with anti-PD-1. Our trial, for example, is for lymphoma or breast cancer or head and neck cancer. And I'm going to say that we believe, having seen some data, that these can be effective. And also the Cornell buddies of ours, Sandra De Maria and Silvia Formente, trying a similar approach in breast cancer as well. Friends at Montefiore, a similar approach in lung cancer, combining FLT3 ligand with radiation as a local Insitu vaccine. So yeah, we believe strongly in it.
Thank you for that. So this next question, I have a visual to share as well. So we have an audience member who wanted to get your thoughts on what you think of lisacell and CLL. There was a presentation at ASH last year. Let me see if I can pull this up for you.
Morgan, while you're pulling it up, I'll just give a word of background, which is that, we think lisacell, so I'll be honest, our hospital is still so far a Kite Gilead, Yescarta hospital pretty much. And then the separate product for mantle cell lymphoma, which is almost the same as Yescarta. But we're looking forward to getting our contracts all signed with Juno or BMS and to get lisacell in our patients. So I can only speak from the expertise of my buddies down the street at Sloan Kettering, who I chat with a lot and who use a lot of lisacell, mostly for DLBCL, also for follicular, not as much for CLL yet. And the result there is that it's an effective therapy, seems to be as effective as other CAR T cells, but really does seem to have less high-grade cytokine release syndrome. It would seem so much so that our buddies are giving these CAR Ts primarily in the outpatient setting, in the clinic, and not in the hospital. And that is a game changer. So it's a very big deal. So overall, very excited about lisacell's, preserved efficacy and seemingly improved safety, it would seem. In terms of CLL, this is a tough space. A couple of these anti-CLL CAR T trials, including this lisacell one that we're showing here, have been ongoing. Some of them as monotherapy, some of them in combination with BTK inhibitors, as is one of the arms here, the ibrutinib arm here. And okay, I'll just speak broadly. CLL, a smaller space with lots of things in it. I mentioned 5 things earlier: chemotherapy, rituximab, BTK inhibitors broadly, multiple generations of them, and BCL2 inhibitors, as well as PI3K delta inhibitors. So all of those things I just mentioned are pretty easy to use and pretty well tolerated. So it's now going to be a small slice of the pizza pie. And I mentioned that, like my 100-year-old grandma, a lot of patients with CLL have a pretty indolent version of the disease and don't burn through all those 5 great therapies I just mentioned. So it's a pretty small slice of that violet colored pizza pie slice now. So it's a tough group to find and to use and to have be healthy enough, because a lot of them have had 5 lines of prior therapies, including multiple chemotherapies to use. And overall, we do think that T cells in our CLL patients are overall defective going in to make the product. So it's asking a lot for the CAR T to solve that T cell defective state that most CLL patients have. It is also a little bit harder to pluck out the healthy T cells from blood that is filled with leukemia, because these patients still have active disease and the T cell is now a rare cell. So it's technically a little bit harder to pluck that T cell out to make the T cell product. And then the efficacy is going to be a little tricky too, because the biggest predictor of toxicity of CAR T cells, cytokine release syndrome especially, is tumor burden. And leukemic patients all have a very high tumor burden, so a little trickier to implement. So I'm a fan of them giving a try, but I think it's a very uphill battle in every direction. Fan of lisacell, cautiously optimistic for its utilization in CLL.
Thank you. I have a question that came in on biospecifics for you too. So do you think that biospecifics could reach the community setting given the level of CRS?
Let me just say there's community and then there's community. You know, there's, I'm not going to pick on anyone, but Tuscaloosa or Chattanooga Hospital, those might be metropolitan areas for all I know, but I just say them because they have funny names, might have a little tiny hospital where there's no ICU available, there's only one ICU bed, and you know, the amount of lymphoma they see nearby in the community may not be a lot, so it may not penetrate every part of the community. But we got, you know, lots of community centers that are doing autologous stem cell transplants, and this is simpler. This will be simpler than that. So just to say, the CRS we saw in the very beginning of these bispecific trials, I don't think will be quite reflective of where the CRS is now, which is much less, partly because we are doing this up titration with almost every version, glofitamab, mosunetuzumab, epcoritamab, they're all working on up titrations. And really now they're all looking at the subcutaneous approach to see if that is safer, because it seems like it might be. And the other way to make it safer is to just treat patients with lower tumor burden disease, which will be easier, you know, once we get past the 5th line patients and work our way up to treating 3rd and 2nd, and then maybe even 1st line patients, because we can cytoreduce them with standard therapies when they have a low disease burden, give them bispecifics then. So yeah, I think they will definitely reach the community. Maybe not Chattanooga or Tuscaloosa. I apologize to audience members from those towns, but yes, they will definitely reach the community.
We have time for one more. Morgen?
Do it. Yeah. So one more question. I'm probably going to butcher the name.
Umbra Elizabeth.
So hang in there with me. Yeah. Umbra Elizabeth was just approved for follicular lymphoma and marginal zone lymphoma. So do you think the safety profile is differentiated, and will it get used?
Yeah, I do think it's differentiated from these 10 years ago PI3K delta inhibitors, which it is sort of a class member, like idelalisib and duvelisib. And, you know, what they'll have to overcome is not so much the toxicities, but the bad branding of the word PI3K delta inhibitor, which we developed from those first 2 members of the class, because they were, it's not that they were pretty toxic, but in, you know, 1/5 of the patients, they had some very frustrating, you know, high-grade colitis and other itides, other itis toxicities. And yes, this Umbra Elizabeth seems to distinguish itself. It does not seem to have, you know, 20% of patients getting high-grade colitis. There might be 3% or some low number that's hard to detect, but it definitely distinguishes itself in the, you know, first couple hundred patients treated for sure. And it can get used if they can get the word out to the community oncologists, because a lot of these low-grade lymphomas are still getting taken care of in the community. So it'll just be a matter of branding and getting the word out.
Josh, that's a wrap. You did it. Thank you so much. Ladies and gentlemen, today I've been speaking to Dr. Joshua Brody. He's Assistant Professor of Medicine, Hematology, and Medical Oncology at Icahn School of Medicine in Mount Sinai. He's also available for bar mitzvahs. So thank you. Thank you once again for joining us today.
Neil, good to see you. Morgan, thank you guys so much.
Thank you guys. Take care.
Bye-bye.